Hello. I want to thank the organizers for inviting me. This is an aging meeting, and you all know way more about aging than I do, even though I'm probably older than all of you put together. We work on hibernation, which is a phenomenon in mammals and other animals where metabolic rate drops and time stops. These animals can extend biological time. They live for longer than they should be, but they live in hibernation, a state where they have 1% or 2% of the metabolism going. You can read about the phenomena of hibernation and our papers by going either to our website, which is listed on this slide, or by going to PubMed. You can go to PubMed and put in Storey KB, and you can find the papers that are coming out and that are out already. Hibernation is a type of a stress. These animals live a warm, fast, completely awake lifestyle, just like other mammals do, but then they have a hidden adaptation where they drop their metabolic rate and they're a different animal. Under hibernation conditions, you drop your use of fuels, you drop your transcription and translation. There's complex physiological changes where their body temperatures can fall depending on the outside temperature. Their heart rates just drop to almost nothing, and they don't use up oxygen and they don't breathe very well. I work on the metabolic part of it, although the papers you can see have a lot of physiological parameters in it. You sometimes see different words. So for mammals, the word is hibernation, but diapause is used for insects and other invertebrates. Estivation happens in the deep desert. Animals dig themselves under the desert and sometimes don't come up for years and years, and they're little sort of dried down, crispy critters without any metabolic rate. Brumination is a word you may have heard. We don't use it anymore. It's for lizards and turtles. When you look at mammals, the stem mammal could obviously hibernate. We think the closest animal to a stem mammal is tenrecs. They're from Africa, from Madagascar. They drop their metabolic rate for long periods of time. They're sort of in hibernation all the time. An animal, a mammal like that, lived with the dinosaurs and sort of slept during the comet asteroid attack. Every group of mammals have some that can hibernate, but a lot of them have forgotten. So think of this as a basic mammalian phenomena that came to us from lizards. In evolutionary time, we used to be anapsid lizards. And you're not on this chart. Humans can't do this. You've opted for breathing all the time. You've opted for staying warm. You've opted for, if the weather goes bad, you build a house, not really hibernate. But when you look at animals as divergent as the little brown bat, which we study, lemurs, which we study, and ground squirrels, they simply live longer. They have a phenomena then where they've somehow hidden themselves away from aging. And so when I read the aging literature, I look to see what's in it, and then I try to translate that to how hibernators live. When you look at what happens, aging seems to me to have a big energy metabolism base and activating cellular protective defenses. This is true no matter how you look at aging in model systems, C. elegans or Drosophila, or mice. And in the longevity models, you can manipulate the phenomena, you can manipulate biochemistry and molecular biology to see how you influence lifespan. What you see again and again, mTOR, the core insulin signaling pathway, SIRTs, FOXOs, and other things. So I'll try to tell you what hibernators do and why it links to me in my mind with respect to aging. So energy metabolism, cytoprotection, heavily regulated during hibernation. We study those and then we look and see, whoa, it's like aging. It's like not aging. Reduced energy metabolism is in all aspects of the cell during hibernation. You stop synthesizing proteins, you stop synthesizing message RNA, you stop extending message RNAs. Message RNAs are held in the cell in little prisons called P-bodies. Ion pumping slows dramatically, and ion leaking slows dramatically. They have ways of changing their membranes. Their fuel use goes down even though they're surrounded by fuels, and their oxygen consumption just falls off the edge of the table. There's plenty of oxygen. They're not anoxic or anaerobic, but they don't have to consume oxygen. The cytoprotective responses we've seen, they're stress responsive transcription factors. Antioxidant genes are turned on. So during hibernation, even though there's low metabolic rate, they've turned on some genes, and they're all antioxidant genes. You do not want free radicals floating around causing you trouble. They bulk up their unfolding protein response, ATF, and there's a lot of pro-survival signaling. The main core of what happens in hibernation, the main thing that we can see is playing with the insulin signaling axis. And this is important in aging as well. This axis you think of as insulin, eating, not eating. In these animals, it's a regulator of cell metabolism and growth and survival. It's heavily regulated. It's the core thing in the cell that's regulated. It's the same pathway in all sorts of animals, all the way from C. elegans, where they have different words for it, all the way up to mammals. All the mammals we studied are the same as humans. You have the same genes with slightly different sequences. You have the same activators and inhibitors. And this evolutionary conservation is something that hibernation relies on. When you look at turtles in hibernation, or frogs in hibernation, or insects in hibernation, insulin is the way to go. This is where nature has decided to twist the machinery and get things regulated. AKT kinases are regulated, mTOR and mTOR kinases, FOXO transcription factors, PTEN, PTEN, and other things above the pathway, as well as SIRTs. So things that you know are linked to longevity phenotypes are regulated in hibernators. The thing is, you study longevity, which is like days, weeks, months, years. Hibernators turn these on and off during hibernation. It takes them 20, 25 minutes to hibernate down, and then they warm up and wake up in just a matter of minutes. They regulate this pathway in an acute way to get really chronic responses. So regulation of mTOR is a big thing. It's a very detailed pathway. It's cored with mTOR, Richter, and Raptor, and then it has downstream events. These are all seen in hibernation. So there's way fewer papers in hibernation. You don't have to worry about that, but it's like longevity. This is the pathway where everything goes on. When you look upstream of mTOR, it's PTEN. It's a molecule that changes during the hibernation cycle. So at the bottom of this slide, you can see a hibernation cycle. They're warm. Their metabolism is normal, EC, control, euthermic. Then they plunge off the edge. Their metabolism falls, and it falls and falls and falls. Even at high temperature, it falls. So if you have a ground squirrel on your desk, its metabolism will turn off even though it's as warm as your desk. Some of the animals go all the way down to 4 degrees, and their metabolic rate is just a fraction of normal. Then they come out. A couple of hours, they're on their way out. P10 is regulated on the right-hand side of your slide, not with message RNA. When you're hibernating, you do not have a lot of energy. You can't make and break down messages. It's phosphorylation. Simple, cheap, quick, reversible phosphorylation. When you look at the activity of this enzyme, it has an activity underneath that receptor complex. There's a huge change. It changes because the kinetics change. So when you isolate the enzyme and do the enzyme kinetics, you find that it changes its kinetics, it changes its activity, and you can see that the amount of protein is the same, but the activity changes, P10. So there's a reduction of phosphorylation, catalytic activity changes, its structural stability is altered, and it reaches out not just to the insulin signaling pathway, but the kinases and phosphatases that are active here also do pyruvate dehydrogenase. Pyruvate dehydrogenase changes dramatically in hibernators in order to shut down their fuel use. So there's a nice link from the top of the pathway all the way reaching into the mitochondria and just cooling their jets, just really turning off their metabolism. Also, upstream in this complex pathway, there's PRAS40 and TSC complex. There's a regulation of these complexes during hibernation and reduced phospho-mTOR. So every time we study control, entrance into hibernation, early and late hibernation, and then recovery from that, interbout arousal, you see this huge effect during late hibernation. You see a phosphorylation and a dephosphorylation. There's a reduced phosphorylation of 4-EBP, S6 protein. This inhibits this cycle. It reduces the activation, and translation also decreases. So by this simple mechanism of taking some kinases and dealing out some phosphates, you can turn off insulin pathway, and then you can re-regulate translation. It is readily reversible. When you take an animal in hibernation and you lift it up, it's really cold, like ground squirrels are cold like ice. They begin to shiver, they begin to shake, they begin to increase their metabolism, boom, and they're on the way to coming back to being euthermic, interbout arousal. mTOR reduction and lifespan appear to be linked in a number of very excellent and thorough studies during longevity studies. I know that people that study longevity may even reach out now to bats, because bats seem to be a longevity model, and now they're looking at why a flying bat that migrates lives for 2 or 3 years and a hibernating bat lives for 40 years. But when you look at your models, when you look at your well worked out genetically regulated molecules, mTOR comes up all the time. Phosphorylation of AKT, mTOR decreasing significantly, and it happens in a lot of different tissues and in every species. You cannot find a mammalian hibernator that doesn't do this, including the stem hibernator, these weird tenrecs. Tenrecs are armored mice that still live in Madagascar. mTOR, AKT regulation during this hibernation period. The overall conclusion is that this is a core pathway. PTEN down to PRAS40, mTOR itself, and then downstream of mTOR, you want to not be making new proteins. So at the bottom of this slide, you can see a little bit. I mean, I can't tell you a lot about it, but there's energy conservation, there's reduced protein translation. The protein translation that occurs in the brain of hibernators is 0.04% of normal. Now, this is hard for your brain. So turning off a mammalian brain is really quite a trick. And they managed to do this and then recover from it. So the brain is a really interesting thing to study just because of this turning off. Most of the studies of hibernating mammals are for transplantable organs. So the brain's so far not transplantable. I can't just cut off your head and move it. Kidneys, hearts, livers, transplantable organs are the focus, and they all do this mTOR trick. The translational arrest is helped by polysomes going away. So under normal conditions, a ground squirrel or a bat is just like you, so it's a tenrec, and under hibernation conditions, they just dissipate the polysomes. The messages are hidden away. The polysomes are still there, but they're there as monosomes. Boom. Phosphorylating EIF2 alpha is a known mechanism. And it's very, very interesting for us to study because it's a horribly complex pathway, 50, 60 different targets, and yet it seems to go ding. If you look at phospho EIF2, you can tell if an animal is hibernating, even an animal that you don't know yet. Transcription factors are involved, several of them. FOXO, which the C. elegans people call DAF, is really critical. It's linked to transcription of a lot of genes to do with longevity. It then regulates pro-aging genes down, and it's also involved in free radical generation, metabolism, and suppression. So you'll see FOXO changed. It changes in all hibernators in almost all tissues. Some tissues have mTOR. Some tissues have FOXO. All tissues do part of each of those, but it's not exactly the same. There's tissue-specific responses. This brings us to SIRTs, which are now really a big deal. They are involved for us in epigenetic regulation. So there's very strong epigenetic regulation during hibernation. And we see it in readers now, writers, and erasers. SIRTs were the first ones we looked at. This was sort of establishing work for us with Drew Rubel, who should have continued in science, but he, of course, went into medicine for the richness. Most of my students end up in medical school. SIRTs are really important. They have to do with calorie restriction. Our animals are calorie restricted. They never eat, but it also has to do with this extension of biological time. The epigenetic markers, DNA, RNA, proteins, histones, becoming a big deal and sort of dominating our lab now from Drew's work. When you look at a summary then of what we find in hibernation versus what I see in longevity, whoa, quite good. Insulin signaling, FOXOs, SIRTs, and other epigenetic antioxidant enzymes. You've got to keep free radicals from running amok. If you're 37, if you're 5, if you're awake, if you're asleep, even in hibernation, you've got to do that. And it seems to be linked in a lot of ways to FGF21 in hibernators. So that's what I have to say about hibernators. You can see the papers, you can see everything that we've done, and there's a bunch of new papers always that are on PubMed. This may interest you, but it's a sign that the work you're doing in longevity is really important, and it's also critical to thousands of other species in the real world. So this is it. This ends my show. Thank you very much. I actually don't know how to turn off Zoom, so we'll have a little bit of a problem here. Thank you very much. Have a good day. No, I can't turn it off.